So, let's get started with this. I'm going to step you through it. Again, the words are on the screen. You can read those at home. I'm not going to just read the words. I'm going to actually explain it to you. So we already talked about the fact that aerobic respiration is made up of glycolysis, pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation. Collectively, that's called aerobic respiration. You need oxygen for that. Anaerobic respiration, same exact steps in anaerobic respiration. Same exact steps. The only difference is you don't use oxygen in this one. Fermentation is glycolysis followed by fermentation. So if you notice, glycolysis is in common in all three of them. Did you notice that? Mm-hmm. In all three events, it starts out with glycolysis. The idea is, are you going to do this? Are you going to do this? Or are you going to do this? Well, it depends on which bacteria you are. And this goes to this slide. If you're an aerobic respirer, if you do aerobic respiration, you probably are one of these three. If you're an obligate aerobe, you're going to aerobically respire. And it's actually mandatory. I must aerobically respire to survive. If you're a microaerophile, you're able to handle low levels of oxygen. You aerobically respire too. But you cannot handle atmospheric oxygen. Remember that. The amount of oxygen in the atmosphere is actually toxic to these guys. So microaerophiles can only handle low levels of oxygen. A facultative anaerobe is an anaerobe that is flexible. It can use oxygen when you give it oxygen. But if you don't give it oxygen, look at it. It has other ways of surviving. A facultative anaerobe can do aerobic respiration, can do anaerobic respiration, and it can also do fermentation. That's why we say it's flexible. So you have to know what each one of these can do. Obligate aerobe, only aerobic respiration. Facultative anaerobe, any one of these. A microaerophile can do aerobic respiration, but only under low concentrations of oxygen. High concentrations of oxygen, like atmospheric oxygen, kills it. Some microaerophiles can also do anaerobic respiration. If you're an aerotolerant bacterium, aerotolerant means I can tolerate the air, the oxygen, but I don't use it because I only ferment. An aerotolerant only ferments. The oxygen does not kill it, but the oxygen is not used by it either. Okay? All right. Let's go forward from here. You guys tell me, which bacterium is drawn on the screen? Gram-negative or gram-positive? Gram-negative. What tells you that?
Cell membrane.
The cell membrane?
The outer membrane.
The outer membrane and the thin cell wall. Very good. So I decided to draw a gram-negative bacterium here. Okay, the process begins with glucose entering the cell. Here it is. Glucose enters the cell, usually facilitated diffusion, because it's too large to do simple diffusion. So facilitated diffusion, it enters the cell. Now it's in the cytoplasm. It turns out that cellular respiration in prokaryotic cells is actually easier than cellular respiration in eukaryotic cells. Why? Because you don't have to deal with the mitochondria. There's no mitochondria that you have to deal with, like in your cells, like in yeast cells, like in amoeba, for example. All eukaryotic cells have a mitochondria, and cellular respiration is compartmentalized, meaning some of it happens in the cytoplasm, but the rest of it happens in the mitochondria. You don't have to worry about that in bacteria because you don't have a mitochondria. That means all of it happens where? In the cytoplasm, except the last part. The last part of cellular respiration in prokaryotes happens in the cell membrane. That's called the oxidative phosphorylation part. We'll talk about it, you'll see it. But most of the event, thank God, happens in one location, the cytoplasm. Again, watch the YouTube videos on eukaryotic cells, please, and compare it with this one. And you'll realize that it's actually much easier in prokaryotes because it's all happening in the soup of things. In the soup of things, in the cytoplasm. So the glucose is a 6-carbon molecule. The next step is to break it into 2 molecules of G3P. You literally, all you did was this. A 6-carbon molecule, add some energy, it breaks into 2 G3Ps. That's it. Each finger is a carbon. Watch, 6-carbon molecule. Add energy, it breaks into 2 G3Ps. Did I lose carbons? No, because if I lost the carbon, that means I would lose a finger, right? This would mean I lost a carbon. This means I did not lose carbons. Well, follow the carbons on the screen. 6 carbons, 2 molecules, 3 carbons each. I did not lose carbon. All I did was initiate glycolysis. Glycolysis, the breakdown of sugar. Glycolysis. And you're going to break down the sugar, 6-carbon molecule, into 2 G3Ps. So far, so good? The interesting thing is to initiate this process, you need 2 ATPs to start it. So some students stop me here and they say, hang on a second. I thought the whole idea was to make ATP. I agree with you. The whole idea is to make ATP. Why are you using 2 ATPs if you're trying to make ATP? That's an awesome question. I have a good answer for you. July the 4th comes around and you throw a party at your house. Come over for hamburgers and hot dogs. I'm throwing a picnic, a barbecue. Come over. So you buy all the hot dogs and the hamburgers and you forget the sparker. Without the sparker, can you light the barbecue? No. So your guests are going to be very disappointed because all you'll have is raw hamburger and nobody likes to eat that. Look at that. Give me the spark. I'll have a fire. See, when you light the sparker over your barbecue, what do? You light the fire. This is the spark. The rest of cellular respiration is the fire. So it asks you, give me a little bit of energy, I'll give you a lot. Give me 2 ATPs, I'll give you 38 at the end. I mean, think about it. If I ask you, give me $2, I'll give you 38. Would you take me up on that? I hope you say yes, unless you don't trust me with your $2. I hope you say yes, because you'll be making money. A lot of money, actually. I wish my bank interest was like that. I'd be rich. $2, I'll give you 38. Nice. But our banks are not like that, unfortunately. So that is called, this phase of glycolysis is called the energy investment phase because you're investing in it 2 ATPs. That's the spark. The rest of it is the fire, the payoff phase. So the next step is that the glyceraldehyde 3-phosphates will become pyruvates. So this is equivalent to doing this. The 6-carbon glucose spark G3Ps. Change to pyruvate. You're done. That's glycolysis. Let's do it again. The 6-carbon sugar spark, it breaks it to 2 G3Ps. Change to pyruvate. We're done. All of this is happening in the cytoplasm. We did not lose any carbon. Did I lose any fingers? No. So we did not lose any carbons. So, at the end of glycolysis, what do you have? You must tally things up. At the end of glycolysis, you have 2 pyruvates. Now, you also have other things. Besides the fact that you use 2 ATPs, to make the G3Ps. When the G3P became pyruvate, look what happened. You produced 2 ATPs. You produced 2 more because this G3P changes to pyruvate like this G3P changes to pyruvate. So you now produced 4 ATPs. You used 2, but you produced 4. So the net gain is how much? 2. 4 minus 2, you gained 2 ATPs. And how are these ATPs made? By substrate-level phosphorylation. And I talked to you about what that is before, but I'll define it in words. It's simple. The simple transfer of a phosphate from a substrate to ADP. We can write it on the board. You can focus over here, Sarah. ADP plus a substrate with a phosphate attached to it. This phosphate will move to this ADP, and this substrate will become a product. So this reaction, the substrate will lose the phosphate to ADP to make it ATP, and the substrate will change into a product, whatever that is. Obviously, there's an enzyme involved here. So this is phosphorylation at the level of a substrate. The substrate has a phosphate, and it will move the phosphate to ADP to make ATP. That's the easiest way to make ATP. Unfortunately, it's not the most common way. The most common way is oxidative phosphorylation. That's the tough one. So 4 ATPs are made in glycolysis by this process. I'll say it again. The simple transfer of a phosphate from a substrate to ADP to make ATP. That's it. All right, now the net gain is 2 ATP. Now in this process, you're also loading 2 U-Haul trucks, 1 for each one. Remember, I called the NADH a U-Haul truck. There's a reason behind that, and you'll see that later when we tally things up. This is an empty U-Haul truck. That's the oxidized form. This is the loaded U-Haul truck. It's loaded with electrons and hydrogen, not furniture, and it's going to be the reduced form. So now I have 2 pyruvates at the end of glycolysis. I have 2 NADHs, and I have a net gain of 2 ATPs made by SLP. Now a decision has to be made. What do I do with this pyruvate? Well, one of 2 options. Either ferment it or send it to cellular aerobic respiration. We're doing aerobic respiration first. So if I don't have oxygen, I will ferment the pyruvate. If I have oxygen, I will pyruvate oxidize. I will break it. I will break the pyruvate and cellularly respire it to get ATP out of it. So listen to what I'm trying to say. The fate of pyruvate depends on whether oxygen is present or not. That's it. I am not saying that oxygen is involved in changing pyruvate into anything. I'm not saying that. I'm saying that pyruvate has to make a decision. What do I do now? Do I go to pyruvate oxidation, the Krebs cycle, and oxidative phosphorylation? I call that the long movie. Or do I do the short movie, fermentation? The decision is made based on the presence or absence of oxygen. Without oxygen, pyruvate will ferment. Into what? We'll see. With oxygen, the pyruvate will go through the full movie. Pyruvate oxidation, the citric acid cycle, also called the Krebs cycle, followed by oxidative phosphorylation, the full movie. But why? Why do you have to get rid of pyruvate? The difficult part right here. Listen, please. You must get rid of pyruvate. If you want to keep glycolysis moving, you must get rid of pyruvate, because there's this weird thing about enzymes and chemical reactions that we learn in Gen Bio. And that weird thing states this: when the product of the reaction builds up in concentration, it will shut down the reaction that made it. So, if you don't get rid of pyruvate, it will turn off glycolysis. Hang on, wait a minute. This bacteria is surviving off of these two ATPs. You shut down glycolysis, it's dead. You see that? The bacteria is surviving off of those limited ATPs that they're getting from glycolysis. So please, it says, please, please, please don't shut down the glycolysis, please. Because if you shut it down, I'm dead because I won't have ATP. Solution is get rid of the pyruvate. One way or the other, ferment it or send it to pyruvate oxidation. I don't care how you do it, get rid of it. So look, you must keep glycolysis moving for the cell to survive off of the net gain of 2 ATPs. To do that, you must get rid of pyruvate. So dilemma number 1, the cell must get rid of pyruvate. There's another problem. To keep glycolysis moving, I have to have enough empty trucks. I can never run out of NAD plus, because if the first second I run out of NAD plus, this one shuts down. So you can shut down glycolysis one of 2 ways: run out of the NAD plus, or don't get rid of pyruvate. It will turn it off, and the cell will suffer. It will die. So look how amazing your cell is, and bacteria. It has 2 problems. It came up with one solution. It realizes something. Hang on, wait a minute. To get rid of pyruvate, I need electrons. Oh, I just loaded those. I just loaded those electrons. So I'll just unload them again. See? The glycolysis loaded the electrons thinking that the truck is going places. It's like you're loading your furniture thinking that you're moving out. And your real estate agent calls you on the phone and says, no, you're not. The new house is not ready. You're not moving out. Unload your furniture. See? The moving out is, I have oxygen. See, you load your NADH thinking that you have oxygen and your delivery truck is going places. But you realize, I don't have oxygen. Then I'm not going anywhere. Instead, I'm going to unload the electrons, give it to pyruvate to change into something else. Watch this now. When I do that, I solve the pyruvate problem. But I also solve this problem. Because when I empty the truck, what do I have now? I have an empty truck. You see that? Your cell, bacteria, it has 2 issues to deal with. I gotta get rid of pyruvate, and I also have to always have empty trucks. It figures out one solution that covers both problems. To get rid of pyruvate, I take the electrons off of the truck that I just loaded, give it to pyruvate to ferment it. Meanwhile, what that means is I made empty trucks again. So what happens now is I'm getting rid of pyruvate and glycolysis keeps moving. The new glucose comes in, 2 ATPs. Another glucose comes in, 2 ATPs. It never shuts down. Thank God. Otherwise, the first time it shuts down, you're done. No ATP, the cell dies. So, the first option that we're going through is fermentation. Come on, one more. The first option that we're going through is fermentation. So the pyruvates realize that there's no oxygen. I gotta get rid of the pyruvate. I'm gonna take the electrons, load them on the truck. See, I'm using the NADHs now. These 2 NADHs are these 2 NADHs, and I'm consuming them. They're not going anywhere. They're not delivering anything. They normally would deliver the electron somewhere, but not these 2. During fermentation, I'm actually unloading the truck. It's sort of like you load your U-Haul to move, then you get a phone call, no you're not, so you unload it. Then you load it, then you unload it. See? You load it, then you unload it. You load it, then you unload it. So you'll always have empty trucks. So when you unload the trucks, you now have these 2 empty trucks that can feed back and keep the glycolysis moving. But meanwhile, what are you doing? You're getting rid of the pyruvates. Look at that. One of these options here. If you notice something, some of the things that we did today in lab fit this here today. Depending on which bacteria you are, you will ferment either this way, this way, this way, this way, or this way, and there are other alternatives too. Some bacteria ferment the pyruvate into ethanol. That's it. And carbon dioxide. Some bacteria ferment the pyruvate into lactic acid. We're like this one. Animal cells make lactic acid, fermentation. All right, some bacteria take the pyruvate, make lactic acid and ethanol and carbon dioxide. They're called heterolactic. They're kind of like a mixture of things. Some bacteria take the pyruvate and ferment it to 2,3-butanediol. Which bacteria? The ones that are positive for Voges Prosecutor test, the one you did today, the VP test. The red color in the VP test identified your unknown to be VP positive, which means it takes pyruvate and makes 2,3-butanediol out of it. That's what that means. Then a lot of you guys had bacteria that was MR positive, except you guys, am I right? I can't remember yet. So the MR positive means your bacteria is actually making mixed acid fermentation. It's producing a whole range of acids, not just lactic acid. So this is what we learned here today in the lab applies to what we learned in lecture here. So different bacteria do different things with the pyruvate, but the ultimate goal is to do what? Get rid of it, ferment it. They just ferment it differently. Again, we're like this one. Do you notice something about the other ones? They all produce what?
Carbon dioxide.
That gas that you will see in the Durham tube. So the Durham tube has the gas bubble. It's most likely carbon dioxide gas, or in this case, it could be other gases as well, like hydrogen and stuff. Okay, so we covered glycolysis, and we covered what happens in the absence of oxygen. It will ferment the pyruvates, and we now know why it ferments the pyruvate. To get rid of it, to keep glycolysis moving. Sounds good? Because then the bacteria is surviving off of the net gain of 2 ATPs until the oxygen comes back. Now we're going to talk about the scenario where you do have oxygen. What happens when you do have oxygen? Now the fun begins. So a while